Hi, today we are going to discuss on the topic. I think most of you, not most of you, every one of you will be aware of this topic, that is blood. So, what is blood? I hope you all know that it's red in color. When we get a wound, blood just oozes out of that damaged area. So you know what it is, what the function is, and everything. But here we are going to discuss about the composition of blood, the various cells present in blood, the fluid part, about their functions and whatnot. So first, about a brief introduction of blood. So, blood is called a fluid connective tissue. So, blood is called a fluid connective tissue. And why is it called a fluid connective tissue? Because the blood cells, that is RBCs, red blood cells, WBCs, that is white blood cells, and platelets, together called the blood cells, are immersed or embedded in this fluid medium or the fluid matrix. That's why blood is called a fluid connective tissue. Now, this fluid has many other terms too. It is called the fluid of life because it transports oxygen from lungs to tissues. The tissues utilize them for their energy processes or for producing energy and releases carbon dioxide that is transported back into the lungs. It is also called the fluid of growth because the nutrients that are produced or formed after degradation of various materials in the digestive system are taken into this blood or absorbed into the blood, and also the hormones that are released by the endocrine glands are transported to various target organs or cells through this same fluid material. It is also called the fluid of health because it protects body against various diseases and also gets rid of waste products or unwanted substances by transporting them to various excretory organs like kidneys, etc. So, from this itself, you can understand the importance of blood. Now, talking about the physical characteristics of blood, its color. You all know what the color of blood is. It is red in color and it is opaque. It is red in color due to the presence of the pigment called hemoglobin. Volume. An average adult human who weighs about 70 kilograms will have almost 5 to 6 liters of blood in his body. Viscosity, that is the thickness of blood. It is 5 times viscous than water. And telling about its pH, that is 7.4, which is slightly alkaline in nature. A neutral pH is 7 and a pH towards the acidic end is below 7. So here the pH of blood is 7.4, that is slightly alkaline in nature. So these are the physical characteristics of the fluid connective tissue called blood. Now, the next is about the composition of blood or what are the components of blood. So blood is mainly divided into two parts, one being the plasma, which is the fluid part of blood, and the other being the formed elements or the blood cells. The plasma contains plasma proteins and formed elements are RBCs, WBCs and platelets. RBCs means red blood cells, WBCs white blood cells and platelets. So this is the composition of blood. When blood mixed with a suitable anticoagulant, that anticoagulant means agents that prevent blood coagulation or blood clotting. So when blood, after mixing with a suitable anticoagulant, is centrifuged for about 20 to 30 minutes at a rate of 3000 rounds per minute, that is 3000 rpm, the blood gets divided into three parts like this. The clear fluid part that occupies the major portion of the tube becomes the plasma or is the plasma, and the red colored part that gets settled at the bottom of the tube is the RBC. And in between these two layers we can see a white colored band, a very thin white colored band, which is called the buffy coat, that mainly comprises of the leukocytes, that is WBCs and platelets. So this is what the composition shows, or these are the parts of blood. The same is shown in this picture also. So the major part of blood is occupied by plasma, that is 55% is made up of plasma. Then 44% occupies the RBCs, and the remaining 1% is mainly the white blood cells and platelets. Now, what are the functions of blood? You all know what the functions of blood are, right? We don't need any particular sort of introduction or anything for describing the functions of blood. Anyway, just a brief introduction to the functions of blood, after which we'll move on to the various parts. So the first and foremost function of blood is the nutritive function. What is nutritive function? That is the transportation of nutrients like glucose, amino acids, etc. that are absorbed in the GIT, that is gastrointestinal tract or the digestive system, is given to various organs or cells for performing their activities. So that is the nutritive function. Then comes the respiratory function, that is transportation of oxygen and carbon dioxide. Excretory function, that is transportation of the waste materials to the excretory organs like kidneys, liver, skin, etc. Protective function, as the blood contains WBCs, that is white blood cells, it helps in defense against various microorganisms or various disorders. Then comes the transport function, that is transportation of mainly hormones and enzymes. Maintenance of body temperature, that is it helps in a balance between heat loss and heat gain in the body. Because of the high specific heat of blood, it is responsible for maintaining the thermoregulatory mechanisms in our body. And finally, the storage function, that is blood stores many nutrients like glucose, amino acids, sodium ions, potassium ions, etc., which can be liberated upon various needs of the body and when the body is in scarcity. So these are the main functions of blood. Now moving on to the main topic of discussion in this video, that is plasma and plasma proteins. As we saw in the composition of blood, 55% of blood is composed of plasma, and plasma is the fluid part of blood, or to be more specific, it is the plasma that gives the fluid matrix to the blood. That's why it is called the fluid connective tissue. So plasma is the fluid part of blood, and it is straw colored or slightly yellowish in color. Speaking about the parts or the composition of plasma, it contains 91% water and remaining 9% solids. Now, what are the 9% solids, or what are the solids seen in plasma? 7% is the plasma proteins, about which we'll be discussing in the later part of this video. 1% other organic substances like carbohydrates, fats, non-protein nitrogenous substances, hormones, enzymes, antibodies, etc. And the another 1% of inorganic substances like sodium ions, potassium ions, calcium ions, magnesium ions, chloride, iodide, iron, copper, etc. So these are the main components seen in that fluid part, that is the plasma. Now, coming to the plasma proteins, or the total proteins in blood. The normal value of plasma proteins in blood is 6.4 to 8.3 grams per deciliter, that is DL. Classification of plasma proteins, that is what we have to know in the beginning. So plasma proteins are of three types. The first one is albumin, then comes globulin, and last but not the least, fibrinogen. So these are the main plasma proteins seen in the plasma. And along with that, there are some studies, or in some textbooks you can see that along with this three plasma proteins, there is one more minor plasma protein, that is the prothrombin, which is a clotting factor along with fibrinogen. Now, about the normal values of these plasma proteins. So the first is albumin, which is seen in major number or major quantity. The normal value is 4.8 gram per deciliter, and it ranges from 3 to 5 grams per deciliter. Then globulin, it's just behind albumin, 2.3 grams per deciliter, ranges from 2 to 3 grams per deciliter. And globulin is in turn divided into 4 types as alpha globulin, alpha 2 globulin, beta globulin, and gamma globulin. Among these 4 types, it is the gamma globulin that forms the immunoglobulins, or the antibodies. Then comes fibrinogen, that is 0.3 grams per deciliter, is seen in very negligible amount. And the plasma without fibrinogen, or plasma devoid of fibrinogen, is called serum. That is what is being used for various biochemical tests conducted in the labs. this picture shows the difference between blood plasma and blood serum. So in the first picture you can see that it is an unclotted whole blood. That is, it is the blood mixed with an anticoagulant. When blood comes in contact with any sort of rough surfaces or any surfaces, when it comes out of our body, it immediately clots or it forms a jelly-like substance. It's some sort of homeostatic phenomenon taking place in our body to prevent excess loss of blood from our body. So when blood comes in contact with some sort of rough surface, it forms a clot. So in order to prevent the clot formation, usually when blood is drawn out of our body, anticoagulants are mixed. So in the first picture you can see it is an unclotted blood, that is anticoagulant mixed blood is taken in the test tube and it is centrifuged, and you can see the plasma, red blood cells and buffy coat. But in the second picture you can see it is a clotted whole blood. That means no anticoagulants are added in the blood and blood is allowed to clot. So after the clot formation, when we centrifuge it, we can get a clear fluid and the clotted part settle down. And the clear fluid here formed is serum, because in that fluid fibrinogen is totally absent, because the fibrinogen that was present in the blood has taken part in clot formation. Fibrin threads are formed, so no free fibrin is present in that fluid part, and that fluid part without fibrinogen is called serum. So plasma is the liquid part of blood in which blood cells, nutrients and hormones float, whereas serum is the fluid part of blood without the clotting factors or blood cells. Nothing is there, it's just clear fluid. So once again, the difference between plasma and serum: centrifugation done directly with anticoagulants, but in serum, centrifugation done after coagulation without anticoagulants. In plasma, clotting factors are present. In serum, clotting factors are absent. And plasma given to patients lacking blood cells, and serum is used totally for diagnostic purposes. Now, coming to the functions of plasma proteins. So, the first and foremost function of plasma proteins is already discussed in the previous slide. So, the first and foremost function of plasma proteins, especially fibrinogen, is its role in coagulation of blood or blood clotting. So, through the formation of blood clots, blood loss is prevented. The second function is contribution to viscosity. What is viscosity? It is the thickness of blood. And albumin is the plasma protein responsible for contributing to maximum viscosity of blood. Then maintenance of osmotic pressure. So what is osmosis? It is the movement of water molecules or solvent molecules from a region of lower solute concentration to a region of higher solute concentration through a semi-permeable membrane. And the pressure exerted by the solute molecules on the semi-permeable membrane is called osmotic pressure. And osmotic pressure exerted by the plasma proteins, especially albumin, plays an important role in the exchange of various substances between the blood and the cells through the capillary membrane. And the osmotic pressure exerted by the plasma proteins is also called colloidal osmotic pressure or oncotic pressure. Then comes their role in defense mechanism of the body. Globulin, especially gamma globulins, help in defense mechanism because gamma globulins form the immunoglobulins or antibodies. So they act against various antigens that enter inside the body, and in that way they help in defense mechanism. Then comes transport function. Albumin, alpha globulin and beta globulin are responsible for the transport of hormones, enzymes, etc. Then role as reserve proteins. During fasting or when there is starvation, the plasma proteins will be utilized by our body tissues or cells as the last source of energy. So how do they serve as the last source of energy? The plasma proteins break up into amino acids, and the tissues will utilize these amino acids for energy production. So in this way, as a last resort, plasma proteins help in the maintenance of or production of energy during starvation or fasting. And the last function of plasma proteins is their role in Roulex formation. Roulex formation is actually a property of RBCs, that is the piling up of RBCs one above the other like a pile of coins. So the RBCs, when they are allowed to remain undisturbed, will pile up one above the other. They will not just clot or they will not have any sort of reactions, but they'll just climb one above the other or climb up one above the other and stay like that as a stack of coins. So that's a main principle behind a diagnostic tool called ESR, that is erythrocyte sedimentation rate. And the plasma proteins, globulin and fibrinogen, are responsible for this Roulex formation of RBCs. So these are the main functions of plasma proteins, and this much about plasma and plasma proteins. So I hope you all got an idea of what plasma and plasma proteins are and their functions, and we'll be coming up with the cells or the blood cells in the later videos. So till then, thank you.